To understand human mood, we zoom past the large circuits of the brain. The regulation of emotion happens here, in the microscopic spaces between neurons. In 1992, researchers isolated an endogenous lipid neurotransmitter operating inside these gaps. They named it Anandamide, drawing from the Sanskrit word for bliss. It is the human brain's primary endocannabinoid, directly responsible for mood regulation, anxiolysis, and the suppression of fear memory. But the system has a mechanical bottleneck. Once anandamide binds to a receptor, it is instantly targeted for destruction by an enzyme called FAAH. FAAH hydrolyzes the molecule aggressively, terminating the internal bliss signal almost as soon as it fires. Receptor activity drops to zero. Achieving lasting psychological resilience, therefore, does not require flooding the nervous system with external psychoactive compounds. The biological challenge is solving this precise enzymatic bottleneck. For 10 millennia, indigenous medical systems have utilized a highly specific botanical matrix to solve this exact problem: minimally processed theobroma cacao. Cacao does not alter mood by delivering high-potency exogenous cannabinoids into the bloodstream. Instead, it operates on a principle of endogenous preservation. The plant contains specific compounds that temporarily disable the FAAH enzyme, forcing the brain to preserve the anandamide it is already producing. Mapping cacao's complete four-pathway neuromodulatory architecture explains how these subtle interactions produce a significant shift in psychological baseline. The pharmacological mapping began in 1996 when a team led by Daniele Piomelli published a paper in Nature analyzing the chemical profile of chocolate. The researchers identified 3 specific lipid compounds inside cacao, known as N-acyl ethanolamines. These compounds are not themselves active cannabinoids. They do not bind to CB1 receptors, and they cannot produce a psychoactive effect on their own. Their mechanism is purely structural. They act as competitive inhibitors. When they enter the synapse, they physically occupy the FAA enzyme, jamming its structural active sites. Because FAA is preoccupied, the brain's anandamide is spared. It accumulates in the synaptic gap, triggering a sustained, prolonged activation of the CB1 receptors. This targeted enzymatic inhibition gave neuroscientists the first identifiable biochemical pathway explaining cacao's documented effect on human mood. But optimizing a single microscopic synapse is useless if the active compounds cannot reach the rest of the brain. This requires the second mechanism in the matrix, a methylxanthin alkaloid called theobromine. Unlike caffeine, theobromine does not aggressively stimulate the central nervous system. Its primary physiological function is to act as an adenosine antagonist and mild vasodilator. It physically widens the cerebral blood vessels, significantly increasing the volume and flow of blood throughout the cortical tissue. Theobromine serves as the plant's built-in transit network. It accelerates the systemic distribution of those FAAH inhibitors, ensuring they reach receptor sites across the entire brain. Subsequent research reviews from 2012 and 2018 identified a third layer to this matrix, isolated within the plant's polyphenol profile. Raw cacao contains dense concentrations of flavonoids, dominated by epicatechin, a molecule that readily crosses the blood-brain barrier. Epicatechin operates entirely outside the endocannabinoid system. Once inside the brain, it drives the synthesis of nitric oxide. This split-screen model shows the divergence. While the left side maintains anandamide tone, the nitric oxide synthesis on the right releases markers of BDNF directly into the hippocampus. BDNF is the structural protein required for adult neurogenesis, physically driving the creation of new neurons and dendritic spines in the region responsible for emotional regulation. The matrix is therefore dual action. The FAA inhibitors elevate immediate mood, while the flavonoids construct the dense physical neuroplastic infrastructure required to sustain that resilience long term. The fourth pathway involves phenylethylamine, or PEA. While the lipid and flavonoid pathways drive long-term shifts, PEA acts as an acute neuromodulator. PEA is a trace amine that modulates monoaminergic signaling, specifically the release of dopamine and serotonin. In minimally processed cacao, PEA is present in concentrations sufficient to trigger a brief but intense monoaminergic pulse post-ingestion. This creates an immediate emotional resonance that frames the experience before the slower FAA inhibition and flavonoid pathways reach physiological significance. Comparing the chemical profile of standard dark chocolate against ceremonial grades reveals why the former fails to trigger these four pathways. This comparative analysis from Crozier et al. Tracks a specific concentration gap in the plant's polyphenol load. This chart shows the disparity. Ceremonial, minimally processed cacao retains up to 63.4 milligrams of polyphenols per gram, while standard 60% dark chocolate contains just 24.8 milligrams. The deficit is a direct result of industrial manufacturing. Modern chocolate relies on heavy roasting and alkalization, or Dutch processing, to extend shelf life and neutralize bitter flavors. But the N-acetylethylamines and flavonoids are thermolabile. They break down rapidly under heat, and the high pH chemical washes used in alkalization physically strip out the pharmacologically active compounds. To hit the compound thresholds observed in clinical research, traditional therapeutic protocols require a specific dose, 28 to 42 grams of raw, unroasted, non-alkalized cacao. Commercial chocolate is a pharmacologically deadened fraction of the original plant. The biological outcome depends entirely on how the matrix is processed and preserved. When consumed at therapeutic doses, the four mechanisms of this intact matrix initiate a specific temporal cascade over a 2-hour window. Phase 1 happens immediately. The PEA pulse provides a sharp monoaminergic spike, generating an acute emotional elevation. Underneath that spike, theobromine creates a steadily rising curve of systemic vasodilation, opening the vascular network to deliver the remaining payload. As the PEA fades, phase 3 takes over. The N-acetylethylamines lock up the FAAH enzymes, establishing a long, stable plateau of elevated anandamide throughout the brain's emotional centers. Finally, phase 4 sets in. The flavonoid pathway initiates a slow, continuous rise in BDNF expression, translating the elevated mood into a permanent neuroplastic structure. These compounds do not add linearly. They act as biochemical multipliers for one another, turning a botanical extract into a highly optimized pharmacological network. This precise compound cascade reveals that for 10,000 years, indigenous practitioners were intuitively optimizing human pharmacokinetics. This effect is magnified when stacked with behavior. When paired with triggers like rigorous aerobic exercise, which naturally produces its own surge of anandamide, the cacao acts as a biological anchor, indefinitely extending the elevation that the exercise created. The utility of ceremonial cacao is an emergent property of endogenous preservation. It does not introduce alien compounds to the nervous system. It uses the brain's own molecular routes to sustain its bliss signal.